All right, quick procedure walkthrough for the mitosis comparison lab. All right, so what you're going to be doing is observing two cells, our onion root tip and the whitefish blastiocells. So the first thing you want to do is get both of these images opened up. You can just have them open up in separate tabs. So there's my onion root tip, and there's my whitefish. I got them both opened up. All right, so what you're going to need to do during the lab is completing this chart and finding cells of each one that are in various phases. So I'm going to do interphase and show you guys that one. So here's my onion root tip. All right, so what's really going to help you a lot, guys, is zooming in. So down here at the bottom, I'm going to use my zoom in button. Okay, there we go. And now when I click and drag, I can move around the slide and I can see all these onion root tip cells. Now I'm looking for interphase, and actually most of the cells that we see here are an interphase. So this is an interphase, 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 interphase. And what you remember about the cell cycle is that most of the cells time spent in the cell cycle is during interphase. So it's not a surprise that we find most of the cells in interphase because that's where they spend most of their time. All right, so what I'm going to do is I'm going to find a cell that I think is a really good looking one in interphase. So this one here I think is a really nice looking one in interphase. I can see the nucleus pretty clear, the nucleolus is pretty clear. Here I can see the cell wall because it's a plant, so it has a cell wall. All right, so I'm going to use my master screenshot tool, and I'm going to take a nice screenshot of it. All right, hit copy, right click on it, copy the image, take it back. All right, so I'm going to put that in my plant because that's my onion root tip. There we go, that looks good. And now I'm going to do the same process for my animal cell. So I would go to my whitefish, I'm going to zoom in, and again, I'm going to find one here where I think has a pretty nice clear nucleus and nice clear nucleolus. So I think this one here is pretty good, or maybe this one here. Those are really nice. All right, so I'm going to grab my master screenshot tool. Oh, here we go, master screenshot tool. Grab one of these guys here. There we go, copy it, copy the image, paste it in. There we go. So I got one for my plant cell, I got one for my animal cell, my onion root tip, and my whitefish. Next thing I'll have to do is give a description of what's happening during interphase. And then for this one is an additional step. It says use the highlight tool to indicate the key structures that are visible during this phase. Not all the structures may be visible, so I only need to highlight the ones that are visible. So nucleus. All right, so if they both have a visible nucleus, which they do in this image, I'm going to highlight it yellow. So I would definitely take my highlighter and highlight that yellow. There we go. And then we'll go through the rest of these structures. And if it's found in only the plant, I would highlight it green, only the animal, highlight it red, or if it's in both, again, like I showed you before, we would highlight it in yellow. All right, so that's the procedure going through this. Take your time. If you have any questions, please ask your teacher.